At this point, your sample's been taken out of the box, scanned, and heated in an incubator, but your spit's still in this plastic tube.
It turns out that cells from your cheeks and gums
And white blood cells.
make up less than 1% of your saliva, and all that DNA that we're looking for is inside of those cells.
So what happens now is a pretty amazing process of separating your DNA from the rest of the stuff in your saliva. The first thing we do is something that's a lot like cracking open an egg to get to the yolk.
Except here we're basically using detergents to bust open all of the cells in your spit sample, freeing the DNA that's trapped inside. And we do a quick check to make sure there's enough DNA before we go into the next step called amplification.
Which is just a fancy word for copying.
Copying.
This copying phase takes up to 24 hours to make sure that we've got enough copies of your DNA for genotyping.
And then we do something that might seem a little bit counterproductive.
Chop up your DNA.
Your DNA needs to get broken up into fragments, into little pieces, and then we end up with bajillions of little chunks of your DNA in a tube. You still with me?
Yes. Cracking, copying, chopping, done.
Next, we need to clean up your DNA.
We spin your fragmented DNA really fast to separate all the stuff we don't want, like leftover cell junk.
And after this, we're ready for the next step, staging for analysis on the genotyping chip.
So exciting!
So exciting! See you next time.